All right, everybody, welcome to chapter 3, which is cells. In this lecture, I'm just going to do the first section of chapter 1. So let's take a peek at the learning outcomes. State the basic principles of cell theory, explain how the surface area to volume ratio limits cell size, and summarize the role of microscopy in the study of cells. So let's jump into it. So cell theory has a few ideas that are associated with it, the first of which being all living things are composed of cells. The cell is the basic unit of life. You can't get any smaller than a cell and still be alive. New cells arise from existing cells through various processes of reproduction and growth. So cells are small enough to be measured in micrometers. This little U-looking thing here symbolizes micro, which is 1/1000th of a meter. For a bit of context, human eggs are about 100 micrometers or 0.1 meters, which is barely visible to the human eye. Cells average approximately 25 micrometers, but have a very wide range around this. There's a huge variance for what sizes cells can actually be. That's just an average, but it kind of gives you an idea. So cells average about a quarter of the size of an egg cell, if that makes sense. So cells are pretty small. And why are cells so small? The answer to that question is the surface area to volume ratio. Cells want to sort of maximize their surface area while keeping their volume small, because it doesn't matter how large you are, you're still going to have to do a whole bunch of exchange with the environment. And if you get bigger and bigger in your volume, your surface area does not increase proportionally. So like I just said, with less surface area, cells cannot easily exchange waste or nutrients with the outside world, and they want to maximize their surface area. So an example here that the book uses is it shows a little cube, and if you imagine cells are cubes, which they of course are not, but it just did it this way so that it can demonstrate easily how to calculate these surface areas and these volumes. You see this little square over here that's 1 millimeter by 1 millimeter by 1 millimeter has a surface area of 6 millimeters squared. This 2 millimeter object has a surface area of 24 millimeters squared. Okay. If you were to calculate the volume, you would see that the surface area of this thing is 1 millimeter cubed, and the surface area of this thing is 8 millimeters cubed, or thereabouts, or actually exactly. So the surface area to volume ratio of the smaller thing is 6, and the surface area to volume ratio of this larger thing is 3. And you can see when you have a smaller volume, when you have a smaller object, the surface area to volume ratio is much larger. Even though this is not that much, this one on the right is not much bigger than this one on the left, the surface area to volume ratio is about half of the smaller object. Okay, so that's why cells are so small, is because they're trying to maximize their surface area to volume ratios. So in order to see these things, we need to use microscopes. This is a picture of a compound light microscope looking at some cells, which you guys should have some experience having done now from lab. The next kind of microscope is this scanning electron microscope, which actually uses electrons to be able to visualize things much smaller than you would be able to see just using visual light. And you can see here's a nice scanning electron microscope photograph. And they do look pretty much exactly like this with the setup and the monitor. And typically what you have to do is gold plate things so that you can see stuff with these. And you can see the sort of scale here, how it's quite small, and the picture is very detailed and also without color. So the next type of microscope is the transmission electron microscope, which gives you more detail than or gives you more resolution than a scanning electron microscope. And here's one with all the parts sort of labeled. I've never seen a transmission electron microscope personally, but it looks pretty cool. And so those are the three basic types of microscopes that you can use to look at things that are too small for the human eye to see. And I put this table here just to kind of summarize what these different things can do. So the eye obviously has 0 magnification, but has a 0.1 millimeter resolving power. And when I say resolving power, what I mean by that is your ability to distinguish between two objects. So if you have two objects that are on top of one another, you're not going to be able to tell the difference between the two. Or if you have two objects that are right next to one another and you don't have a lot of resolving power, you're not going to be able to see the difference between the two. So when you move up to a light microscope, obviously your resolving power gets a lot larger. This should be a period, not a comma. Sorry, I'll just leave that there. When you get up to a scanning electron microscope, you are increasing that by 2 orders of magnitude, or 2 zeros beyond the decimal point. And when you get up to transmission electron microscope, that number gets even smaller. You're able to tell the difference between two objects that are even more close together than with a scanning electron microscope. So obviously there's no light, or there's no magnification for just your eyeball. Light microscopes tend to max out at 1,000 times magnification. Even then, 1,000 times is pretty difficult. You have to usually use oil immersion because of the properties of light. Scanning electron microscope has a magnification of about 30,000x. So you can make something 30,000 times bigger when you're looking in a scanning electron microscope. And a transmission electron microscope is 50,000x. So you can make stuff even bigger than with a scanning electron microscope.